today, what I thought I'd introduce is starting with sample collection. So I think it's interesting that Liz asked the question around where to sample, because I think this is a battle that I constantly have. So I'm going to touch on thinking about sample frequency, types of sample collection, storage, and some shipping, and then move on to think about sample treatment, so cleaning up the samples and concentrating them, including the use of standards, so internal surrogates, and then moving on to the analysis section. So I'm going to briefly talk about direct injection as a method of analysis, and also using SPE, LCMS. And what I'm going to focus on is targeted analysis, but then at the end, I'm also going to talk about some recent mass spec advances where we can think about doing some more sophisticated non-targeted analysis. And then lastly, I'm going to hopefully point you in the direction of some useful resources and some methods and some protocols. So I just want to say that the presentation that I've pulled together, so this has been built on a mixture of published literature. So there's a lot of really good research on this at the moment, as well as based on my experience working on sampling and monitoring campaigns. So hopefully I've got a good balance here, but I'll say that all sources and all references should be on the slides, but I'm happy to share any papers or reports if they would be of use to anybody. So I guess the reason why I want to include sample collection, because I think it's important when you're thinking around sample analysis about where the samples actually came from to ensure that they're representative of what you're trying to measure. So I think Liz alluded to this really well. So ideally with a monitoring campaign, you want it to be devised so that it gathers this spatiotemporal picture of chemical pollution. So you would take samples regularly and at a catchment scale. And I've drawn a quick sketch here as an example of a relatively small sampling campaign. So you can see the point of discharge at the wastewater treatment plant. And then this is where you would sample to look at the immediate effect. And then you could ideally sample further downstream so you can evaluate the chemical fate. And what you really want to do is to be able to compare your sample to a pristine site, so typically upstream. And this is where you would have a sample that's free from contamination, and it was used as a background. However, I'm sure that, as most of you are aware, when you're carrying out monitoring campaigns, it's actually quite a challenge to find a site that's free from any form of contamination. Even in the UK, we've trekked into the Yorkshire Dales to try and find a background site, and we still managed to find some contamination. So an upstream site, it's a bit of a caveat just to say it doesn't always mean that it's a clean and background sample. And when you're thinking about your method reproducibility and quality assurance, quality control, what's really important to do is to collect blank field samples. So this is typically, you take some HPLC water into the field and you would treat it in the same manner as you would with your actual environmental sample. And this is just to check for any contamination in the field through sampling all the way through to sample prep and analysis. So now in terms of the actual collection of the sample, for pharmaceuticals analysis, this is quite often done with a grab sample. So you can get grab samples of water from milliliters to liters, and it's suitable for moderate to high chemical concentrations. But what you often find is that it fails to detect trace levels and only really shows this single snapshot of chemical levels at a specific point in time. So an alternative is to do passive sampling, and this collects water directly in situ. And so then you get the accumulation of pollutants over time. And this is what I'm going to go into in more detail in the next couple of slides. But I guess ultimately what you're what you're looking for is to collect samples with sufficient spatial resolution that you can build these concentration profiles and increase your probability of detecting these transient and tricky pharmaceuticals. And I think when you're devising your sampling campaign, this is important to do, but obviously you are then met with the logistical challenges of where you're actually sampling. So where to collect your sample? I think one of the main drivers of this is largely the size of the water body that you're dealing with. So in large rivers, there's more chance of variability in water characteristics and quality of the water across the river. There may be a tributary coming in, for example, from above the sampling point, or you may get a wastewater treatment outflow pipe upstream, and it takes longer for the water in a river to typically mix. So this means this takes longer for your chemical to mix and become this ubiquitous presence in the water. So if you were to take one sample, for example, on the side of a river near a discharge point, then this might not be representative of the whole river stretch that you're interested in. So it's important to bear in mind that this isn't an accurate representation of the pollution load. So the figure on the right-hand side of this, this slide is taken from the supporting information of the paper by Emily Burns, who I worked with on a monitoring campaign in York. And she designed this study to look at chemical variability across a river stretch by taking a lot of repeated samples at 3 points across the river, and then comparing chemical concentrations with target analytes. And it's probably important to point out here that our rivers in York are relatively small. So we found that these were well mixed. And as you can probably see by the charts in this slide, there was little variability in the chemical composition across the river. So what we were able to determine here was that a single sample, whether it be in the middle or at the edge, was sufficient in order to give you this accurate representation of chemical concentrations. So this is, I guess, an ideal if you can do some preliminary work to look at the concentrations and devise your sampling campaign as to where best to sample. But if this isn't possible, what you can do is take water quality parameters to understand if your water is well mixed. And if it is, then this would suggest that there's only a need to take limited samples to get this whole complete picture. And it's just a note really, is that when you're planning your sampling sites, it can be really useful to plan these around access to bridges. So this is something that we've done in the past, as well as looking into the use of boats, for example, if you need to get this stretch across the river rather than just a single sampling point. So I guess when I started doing chemical monitoring, I had this grand idea that I would be using some fancy high-tech equipment, and I could potentially be floating across a river and collecting some wonderful river samples. But in reality, what it ended up being was me finding lots of bridges and pulling up large quantities of water, which is quite hard work, especially in the British rain, as it was mentioned, and lugging up buckets up 30-meter bridges. So this is a grab sampling collection. And when you have limited infrastructure or capacity for this advanced sampling techniques, then grab samples are really useful and they're a cheap option. And as the name suggests, it's literally a case of grabbing some water. So you can do this in a bottle, you could put a bucket in a river. It's literally taking a portion of water for analysis. But most pharmaceuticals and metabolite transformation products exist in our environments at trace levels. And on the whole, most water samples tend to be collected by grab samples, but this, as I mentioned before, only means that you're measuring this instantaneous concentration at a certain moment of time, and doesn't necessarily reflect this average concentration of target substances in the water. And this slide, I think, is really important to highlight how you have to be careful about when you select your time to do your sampling. So the grab samples don't capture this diurnal variation in chemical concentrations. So I've put here a figure taken from a study by Monroe and colleagues published in 2019. And this was a 12-hour diurnal occurrence study. And it was conducted in London between the time of 7 in the morning and 7 in the evening. And they collected water samples during this period every hour. And this sampling site was in the Thames, so central London. And what we know about the Thames is that the water levels change on a daily basis and can be up to about 7 meters. And this typically occurs twice a day. And the river flow is actually relatively small compared with the volume and the tide. So on the plus side, it is relatively well mixed. So coming back to this figure, so the black data points represent the mean of 2 grab samples that were collected, and the grey dashed line is the tide height at the time of sampling. And what you can hopefully see here is that the time of day the samples were taken strongly influences the measured concentration. And probably what's most relevant to our program is that the levels of antibiotics in particular are seen to vary most throughout the day. So trimethoprim and sulfamethazine were the only, the most obvious cases that showed any correlation with tide or water conductivity. And in fact, these almost doubled in concentration with high tides. So I think if you can look at chloramphenicol, the concentrations range between 5 and 25 nanograms per litre over the course of the day. So in terms of data interpretation, it's really important to think about how representative your sample is. So if you take this example, if you were to sample in the morning, this would underestimate your average concentrations. So the next sampling technique I'm going to talk about is passive sampling. Passive sampling has recently become a major tool when you're looking at organic contaminants in surface waters, groundwaters and wastewater as well, in fact. And a whole suite of contaminants have been studied using passive samplers. In addition to prescription drugs, you can also see illicit drugs, pesticides, PAHs using this sampling technique. And the passive sampling really overcomes some of the disadvantages that I've talked about when you're talking about grab samples. Because in theory, it objectively reflects the overall pollution situation of your substances in the surface water. So a polar organic chemical integrative sampler, or POCIS, has been really well used for studying the presence of pharmaceuticals in the environment. And this is a relatively low cost device. It's non-mechanical and it's quite simple in design. So this slide just shows the concept of POCIS in a bit more detail. So it comprises of this receiving sorbent, and it's enclosed between 2 diffusion membranes. And to prevent the loss of these membranes, you encase this within some O-rings, and these are typically made of stainless steel. And you sandwich your sorbent in between all of these rings, and it's placed most often in like a metal basket, which is then left in situ in the field. So the idea is that large substances, so thinking about bacteria, macromolecules, particulates, are excluded from this device, but it can capture your trace contaminants. And there are a number of different configurations for this sorbent material in the middle. Probably the most relevant is the pharmaceutical POCIS, and this is a hydrophilic lipophilic sorbent, and it's called the OASIS HLB, which is made by Waters. And this is a water wettable reversed phase sorbent. And so basically it comprises of this, this polymer that can absorb all of the chemicals. And yeah, so OASIS HLB is probably the most frequently used in this case. And it's been widely used for pharmaceutical detection, including in antibiotics. So at the moment, this all sounds great. But if you're going to use these quantitatively, then they require quite some effort to calibrate them. So you need compound-specific uptake and diffusion coefficients, which require quite a lot of work to generate, and they can be determined both in situ as well as in the labs. And you also have to deal with potential losses of the resin or sorbent. So even though they're encased within this system, sometimes you can lose the resin, and then you need to account for that when you're doing your quantitative calculations. And lastly, the final option I'm just going to briefly introduce is a composite sampler. So like passive sampling, it's effectively collecting the pollutants over time. So you get this more comprehensive picture of contamination. And what you do is set the collection samples at the frequency and the volume you use. So here I just provided the example that we've used before. So it was 24 hours of collection, and every 30 minutes where 50 ml was collected into this big tank. However, the downsides are these are relatively expensive, and I've seen a lot of studies place them in wastewater treatment plants, for example. But if you were to typically leave these out in the field, in a river, then I wouldn't guarantee that it would be there by the time you get back. So potentially some downsides here. And again, if you were going to use it for quantitation, then you need to do some in-field, in situ measurements to get the data and combine this with information like flow so that you can determine concentrations over time. So now moving on to sample cleanup. So you've collected your sample, and what you need to now work out is what to do with it. So the two options that I'm going to focus on are SPE LCMS-MS, as well as direct injection LCMS-MS. So direct injection prep is very simple. In the field, you would filter your sample after you've collected it. So typically you use a glass fiber filter and you put it into the LC vial for analysis. And the filtration step removes the particulates and the bits that you don't need, but retains the chemical within the solution. And there's been a number of studies that have shown that there's quite minimal retention on the actual filter, because I think this was a concern in the beginning. So one study that I'm familiar with looked at about 26 compounds with different properties. So it's acids, bases, amphiters, had chemicals ranging in hydrophobicity, and they found that the losses were less than 5%. So as I said, relatively simple. You take your sample, you filter it, and then it's ready for analysis. And I'm going to talk about the analysis step later. You typically have to inject a larger volume into your mass spec for your analysis, but effectively your sample is ready to go. Whereas for SPE, so this is solid phase extraction, you have to take your samples back to the lab where the SPE process, it cleans and concentrates your sample. So it's a more involved process, but it can take various forms. It can be done online in the mass spectrometer, as well as offline using cartridges. And when I looked into this a bit more for this presentation, I found quite an interesting review on monitoring studies to evaluate the different techniques they use for sample prep. So this was between the period of 2018 and 2020. And what they found were that quite overwhelmingly, classic SPE procedures were most commonly selected when you're thinking about sample prep. So this was mostly cartridges that were used offline, so in about 70% of studies, whereas 16% of studies used different methods, which would include, for example, direct injection. So coming back to direct injection, it's relatively simple in terms of sample prep, and even though it's not as widely used as SPE as a method, I think it's gaining prominence in terms of a reliable method to determine pharmaceuticals in river samples. But on the counter argument, SPE has many benefits. If you have particularly mucky samples and filtration just won't work alone, then SPE will be necessary. As well as if you really need to concentrate your samples, then SPE is something that you can do for that. So just now what I'm going to do is go into a little bit more detail about solid phase extraction itself. So SPE is a form of liquid chromatography. And it's widely used for organic analysis of agrochemicals, pharmaceuticals, and other contaminants. But the basic principle of SPE is that your compounds of interest, they partition between two phases of solid and liquid. So there must be a greater affinity of your compound for the solid phase than for the sample matrix. So your water sample that's collected from the river. So what you want is your sample to come through the cartridge and the chemicals to be retained on the solid phase. And then you would then elute these chemicals of interest with a solvent. So the sorbent that retains your pharmaceutical, your chemical, is encased within this barrel syringe. And after you activate your sorbent, you pass your water through. So this can range up to hundreds of mils. We've passed liters, up to about 1.5 liters of water through a cartridge before. And then, as I said, your contaminants are retained on the sorbent, and the water is then discarded, comes out the bottom of the cartridge. And then you elute your compounds of interest using a few mils of solvent. So this is a relatively time-consuming process, but if you're dealing with trace level analysis, this is really important. And SPE, there are many different cartridges that you could use, but they typically separate your compounds based on the principles of reversed chromatography. And there are a number of different characteristics which I've sort of included here as to the types of target chemicals and the best types of sorbents and SPE columns that might be useful in the future. But typically for pharmaceutical analysis, what tends to be used is an OASIS HLB cartridge. And I just want to introduce this paper here by the Diana Arga group. And I think it would be potentially of interest to quite a number of people working in the program if you haven't seen it before. So it's describing methods for the measurements of antimicrobials in wastewater and surface waters. And they have done a really nice job of validating the various steps in sample prep and sample shipment. So here they talk about SPE methods that might be of use to you, but I'm not going to go into the actual SPE method itself. But what I think is the thing I'm going to talk about is actually thinking about SPE as a method of sample shipping. So performing solid phase extraction after you've collected your sample is great, because firstly, your shipping and your handling costs can be minimized because you're only shipping the cartridges rather than glassware filled with all these liquid samples. And secondly, there's less chance of your samples breaking if they're retained on these cartridges. And as I mentioned in this paper, they talked about validation steps and investigated the stability of antimicrobials in SPE cartridges. And they tested them under 2 different storage conditions that would typically be found if you were shipping these samples. So they had -4 degrees and room temperature, which was defined as around 23 to 27 degrees. So these were defined as worst case and best case scenarios. So the best case would be to ship on dry ice, and the worst case would be to ship at room temperature. And the antimicrobials that they evaluated included sulfonamides, macrolides, quinolones, and tetracyclines. And in general, what you can see is a decrease in analyte recovery over time for the compound classes. So I think this is to be expected, really. The stability of the analytes inside the actual cartridge also differs by compound class. So your sulfonamides and your tetracyclines show much faster degradation than the macrolides and quinolones, which tend to be more stable inside the cartridge. However, I think the most important point to note is that cold storage increases the stability of the compounds significantly. And it shows higher recoveries of the analytes from the SPE cartridges, even after 1 week of storage. So by keeping the cartridges cold during the shipment, the integrity of the sample is ultimately maintained. So if you ship your samples at room temperature, you typically be underestimating your concentrations because you would have this degradation occurring that you're not accounting for. So you have your sample in an LC vial, and this has either been filtered in the field, ready for direct injection, or it's been concentrated and cleaned up by solid phase extraction. And so the next part is the analysis. And when I first started working with pharmaceuticals in the environment, mass specs were incredibly hard to get your hands on, and we used to do a lot of work with HPLC UV. It's really cheap, robust, relatively straightforward. However, when you're dealing with complex environmental matrices with multiple chemicals, then its specificity is one of the drawbacks that you have to consider. So this reason, mass spectrometry became a really big player when you're looking at the presence of pharmaceuticals in the environment. So I guess to try and explain LC-MS, it's probably easiest to split this into 2 parts. So the LC and the mass spectrometer. So the LC stands for liquid chromatography, and it's a separation method. So your sample gets injected into the LC, which uses really high pressure, and it separates the sample components and then introduces these into the mass spectrometer. And this, so mass spectrometer or MS, is the part of the analysis, and this creates and detects ions and allows you to work out what is actually in your sample. And what's important to note here is you can have both LC-MS and LC-MS-MS. So a single MS means that you only analyze your precursor ion that's generated in the source, whereas MS-MS is the combination of 2 mass analyzers in 1 instrument. So the first MS filters the precursor ion, and then you get this fragmentation of the precursor ion with lots of high energy and gas. And then a second mass analyzer then filters your product ions. And really the advantages of MS-MS are that they increase the sensitivity of your analysis. And you can get more structural information about the fragmentation pattern. So tandem mass spectrometry, or MSMS, typically uses something called a triple quadrupole mass spectrometer. And this is what you would quite often see when you're reading papers to do with the quantification of pharmaceuticals in the environment. So the next couple of slides, I'm going to go into more detail about this. So to start with, your sample is injected into the HPLC with flowing mobile phase. And if you were to use direct injection, then typically you would be injecting around 100 microliters of the sample. But if you're using more traditional methods, then it's typically around 10 microliters. And polar compounds, such as pharmaceuticals, can then be well separated on a reverse phase HPLC column. And this is operated under a gradient option normally, where you get aqueous to organic gradient solvent flow. And then your pharmaceuticals, once they've been separated, are then transferred to the ion source. So this is just a quick figure to demonstrate a little bit more about reverse phase chromatography. So this is the column that your sample goes through. And you have your mixture of components and then a stationary phase, which is your column. And the most hydrophobic parts of your sample interact with the column best, and they get strongly absorbed, whereas the least hydrophilic components elute first. So this is how your sample gets separated and how you get the range in times of which your samples are detected. So coming back to this figure, your sample is now in the mass spectrometer in the triple quadrupole, and this is what I'm going to talk about now. So a triple quadrupole mass spectrometer. So this is the MSMS. And here you've got 3 quadrupoles. Your first and your third quadrupole, these act as mass filters. And the second causes this additional fragmentation. And a common sequence here is you get a product ion scan, then you get a precursor ion scan, and then you get a neutral loss scan. And then it's followed by selected reaction monitoring or multiple reaction monitoring. So this is just to demonstrate the output that you would typically get from a mass spectrometer. This is a target ion trace of a spiked pharmaceutical standard. And what you now need to do is to process and interpret your samples. So a major challenge in analytical chemistry is the development of repeated sample preparation methods that give you these acceptable and reliable recoveries that can guarantee the correct representation of your result. So you have to consider the potential for matrix effects. And this is something that's quite widely reported for the analysis of environmental and biological samples. And a lot of this has been linked to the presence of natural organic matter. But from what I'm familiar with, this mechanism still hasn't really been unraveled. But what you're doing is really developing your mass spec methods to minimize the matrix effects. So you can use different methods of extraction, of cleanup, of elution. So filtering is one aspect of this. Solid phase extraction, as we talked about. You can do different liquid chromatography procedures, as well as reducing your elutant flows, for example. However, the problem is when you try and apply these to simultaneous analysis of a whole suite of compounds, you get varying degrees of ion suppression and enhancement. And So there's been a number of methods that have been proposed to compensate for this, using different calibration techniques. So this is where concepts such as standard addition, surrogate monitoring, and various forms of internal calibration come into the analysis. So isotope dilution is a means to compensate for matrix effects, and it's generally considered to be one of the most recommended and most versatile approaches when you're dealing with matrix effects. So when thinking about internal standards, you have to consider a number of things. So your internal standard shouldn't be present in any of the samples analyzed. For example, so if you're using a prescribed drug as an internal standard for a therapeutic drug monitoring campaign, it's not a good idea. It also needs to be similar in the physicochemical properties to your target analyte. So good ideas here are isomers or homologs. And an important point, I guess this brings me to, is the discussion around terminology of internal standard, surrogates, external standard. And I think that the nomenclature around internal standards is not entirely clear. So different people call these different compounds different things, typically based on when they're added to the analytical process. So analytical internal standards can be added as late as just before the injection into the HPLC, and sometimes they can then be called injection standards. And this is basically used to provide a reference of instrument detection variability. However, if your standards are going to be used for correcting your extraction and cleanup recoveries, then they needed to be added as early on in the process. And so sometimes people use multiple internal standards added at various different points to account for all possible sources of variability. So just to recap, if you add your internal standard at the beginning, this is typically called a surrogate. And this is when you can account for errors in sample preparation. If it's added just before the injection and analysis, then this is more likely going to allow you to account for recoveries and instrument variability with the instrument specifically. So typically when you select an internal standard, the rule is that whatever happens to the analyte or your compound of interest will happen to the internal standard. And most often we use an isotopically modified version of the target analyte. And what we want is that we don't want these isotopically labeled internal standards to separate from our analyte when we're doing the liquid chromatography, but we want them to be separated by their mass difference. However, isotopically labeled standards are expensive, and not all analytes of interest have a corresponding labeled standard. So in this case, a number of reports have suggested using a corresponding internal isotopically labeled internal standard, such as atrazine. But there are the options are typically deuterated, carbon-13 or nitrogen-15 labeled compounds. And I just wanted to point out here, when I spoke to Jan recently, he was asking about which isotopically labeled internal standards to use. And I've always used deuterated internal standards. And I think a lot of methods that have been published in the literature use deuterated internal standards. But when Jan asked why, I started to look into the literature a bit more. And in the chromatography literature, it's suggesting that, but in the mass spec literature, sorry, it's actually suggesting that carbon and nitrogen labeled compounds are generally more preferred because you get less of a shift in the retention time in comparison to your analyte of interest. Whereas the deuterated compounds, you get a bigger change. But I think the published methods for pharmaceutical analysis in environmental samples, so the USGS method, the US EPA method, as well as other published literature sources, they all use deuterated standards. And I think what your choice really comes down to is the availability. So when I looked on some internal standard websites just now, I couldn't even see any carbon labeled compounds for my chemicals of interest. So I think availability is a large factor here, but also what can reflect your analyte most in terms of your analysis. So I'm aware of the time, and I shall probably start to speed up to give time for a question or to move on to the next talk. But really the last part of the presentation that I have put together is about quantification. So what you want to do is to use all this analysis to determine the concentrations of your chemicals of interest in your samples, or quantification. And the two methods I'm going to talk about are external calibration and internal calibration. And in external standard calibration, you plot your absolute analyte response against a known analyte concentration. And this creates your calibration curve. So in the example on this slide, I've got a range of calibration standards and then the corresponding peak areas. If you plot these, then you can use the regression to calculate your unknowns when you have a measured peak area. In comparison, when you've got internal standard calibration, so this is when you use your stable isotopes and your sample is fortified with these. So this is along the same lines, but it changes slightly and plots the ratio of the analyte response to the internal standard response. So this is called a response factor against the ratio of the analyte amount to the internal standard amount. So your resulting calibration curve is applied to the ratio of the response of the analyte to the response of the internal standard. And most modern software packages contain algorithms that automate this, but it's important that you check your peaks and these are inspected to make sure that these are integrated properly. And then lastly, what we have to deal with are issues of carryover and contamination. So it's important to have properly validated methods. And I don't have time to go into all of these, but the paper by Luz et al. In 2016 is really nice, talking about quantitative mass spectrometry and the ideas around accuracy, recovery, and precision. And another useful source when thinking about quality assurance and quality control is the paper by Ed Furlong, looking at using mass spectrometry for the determination of human pharmaceuticals in filtered water. And here they go into quite a lot of detail about the different types of QA and QC samples and when these need to be used. So, for example, the presence of a lab reagent blank, so you can check whether there's any contaminants in your sample or carryover, as well as thinking about your field blank samples, for example, that these were the ones that were prepped in the field. So I don't have enough time to go into non-targeted and suspect screening, but suffice to say that this is what I've talked about in the analysis section is targeted analysis. So when you're developing a method based on the determination of analytes that you know and you want to quantify, and these are the ones that you can then analyze and work out the concentrations for. However, more advanced mass spectrometry methods now are available, such as suspect screening and non-targeted screening, which take a more comprehensive evaluation of your sample. So thinking about unknowns and trying to identify those. Okay, so that brings me to the end. So I hope that's been helpful. And I've put a slide up with some additional resources, which I'm happy to share about water sampling techniques and methods that have been published and are well accepted in the field. So thank you very much.